So we are starting with the new chapter called Principles of Inheritance. So it's going to be really about genetics and all the other stuff. Actually. Right. So yes, coming to the questions first. So what do you all know about genetics? Amrita, where are you?
Ma'am, are we doing a new chapter?
Yeah, we have completed the other chapter, right? Last time?
Yes, ma'am. Yes, ma'am.
Yeah, we are starting with a new chapter.
Okay.
Chapter number 7, that is unit 7 in your book, which is genetics and evolution. This is going to be really a huge chapter for us. It's going to be taking a lot of time. And one of the most important chapter, I would say, and really interesting chapter, actually. Right.
Ma'am, are we not doing in between chapters, molecular inheritance and other one?
Yeah, that genetics is going to be all the 3 chapters, no? Principles of inheritance, molecular.
Okay. They've given us separately, so I just asked you.
Yeah, that is one complete unit and it has 3 chapters in it. That's what I said. It's going to be a long chapter, the long unit, what we are going to be discussing. So probably, I guess it would be taking more than a month or so. So anyway, we'll just start with the topic today. Yes, one of the very important chapters where you get questions for competitive exams as well. So we have to be targeting this chapter more. Yes. So what is genetics? Come on, tell me. Just your idea about genetics. Yes, Kiran Sarana.
Like genetics is the study of heredity.
What is heredity?
Heredity means like a generation wise generation is the same characters.
Okay.
Like study of heredity and the variations. And
what is variation? What's variation? Come on. Yes, Hari. Any idea what's variation?
Ma'am, genetics is related to gene, chromosomes.
Yes, good. And apart from that? Okay, what is gene? What is chromosome? Where are they located? What's its structure? What's it made of?
Ma'am, gene carries hereditary information from one generation to other generation.
Okay, where is it located? Where is gene? What is gene, first of all?
DNAs, RNAs together, nucleosomes.
Oh, wow. You are making me to ask a lot of questions by your answers, actually. Okay, anyways, fine. So then it would be a question answer session rather than me teaching you. Okay, anyways, fine. So what is genetics? So we are going to be talking about a lot of interesting aspects. I would like all of you to ask me a lot of questions. That is going to be your job in this chapter. So it's a branch of biology, as all of you said, this is the definition, which deals with mechanism of inheritance and variation. Okay, so what is now inheritance? So the question is, what is now inheritance? So it is telling that it's a mechanism of inheritance. So there is some mechanism through which certain characters are transmitted from the parents to offspring, a generation after generation. So we are going to be dealing with that particular mechanism actually. So mechanism of inheritance and variation, that's what is genetics here, right? So biological heredity from parents. So what is the difference between inheritance and heredity? Who's going to tell me? So are these words same or is it going to be a different thing? So what is inheritance? What is heredity?
It's just a guess. Inheritance might be from one generation to the next generation alone, but heredity is sequence of generations where one character is passed to every single generation.
Very, very good. Right, clear answer. So inheritance means the transfer of certain characters from one generation to another. So the flow of information through generations So the information is flowing from forefather to father, father to son, son to grandson, and so on. So that flow of the inheritance is going to be heredity. So heredity is the basis of inheritance. Inheritance is going to be the basis of heredity, right? Okay. So, yes, once again, Sudarshan is joining. Okay, Sudarshan, good evening. I just want you to switch on your camera. Thank you, Sudarshan. Fine. Okay, so Sudarshan, we have started with a new chapter called genetics, and we are just looking to the first slide of that chapter, right? Okay. So what are the different characters? Okay, now the question is really here. Please understand what I'm going to speak, so you will answer my question. Well. So we say that characters, right? So we say characters. What are those characters? Excuse me. So it's going to be the hair color, the skin color, the eye shape, the eyeball color, the hair texture, the hair length, and all these things. So how is the character stored in the DNA? In what form is it stored? So we say it is being transferred from one generation to another. How it is transferred? By means of this genetic material. So how does the baby, or where does the baby get the genetic material from? So that was the last chapter we dealt with. Am I right? We did reproduction. Yes? So what's happening in reproduction? What is the only thing that is getting transferred? So it doesn't reproduction, does the parent get the whole sorts of legs, hands, everything? No, just the nucleus is fusing. Only the nuclear material is fusing here. So it's obvious that the DNA is the one that's carrying genetic information. In what form is it carrying? In what form is it carrying? How does the DNA decode? What's there in a DNA?
Ma'am, DNA have nucleotides, ma'am. The arrangement of nucleotides is decoded by the mRNA present, which like codons are present. Ma'am. So on decoding that, you get the genetic information.
Okay, so in what form is the character expressed? You're right, the flow is like this. So it's decoded as mRNA. In what form is it expressed? Whatever character we have, in what form is it expressed? Yes, proteins. Yes. So usually this is what we call a central dogma of molecular biology. Just one second. One second. I'm really sorry. I have a dry throat actually. So the central dogma of molecular biology is this. So from DNA, it is mRNA. So mRNA. So from mRNA, it is protein. Okay, so PTM I just mentioned as protein. So any character in our body is expressed in a form of protein. So skin color is due to melanin. Hair growth is because of keratin. So I can tell everything. So eyeball and eyelids, everything is form of some protein that's being expressed in our body. So that is what is being stored in the DNA. So imagine, we'll be doing a lot of things in molecular basis of inheritance. I will let you know at that time, but generally understand. So it is very beautiful how we store such a huge information in a small pen drive. It's like that all the informations are stored in the DNA in the form of nucleotides. So it's so beautiful and fascinating how lots and lots of information is stored in such a small cell in our body, and every cell in the body has the same information. Now, children, tell me. So we'll be talking about this in detail later part of the chapter. The question is, every cell in your body, please get the question. Every cell in our body has the same DNA, same chromosome, right? All of you agree with that? Every cell in your body has the same genetic material in the DNA, right? Yes? Okay. Now, tell me why only your stomach produces acid and your eyes doesn't produce the acid, right? Your stomach only produces HCl. Your eyes doesn't produce it. or your eyes only produces tears, your stomach doesn't produce it. How is that happening? How is that message? Where does that gene get that message from? How is it possible? See, every cell in your body has the same chromosomes in it, right? Yes, yes. So how is then these genes are like, how only particular thing are expressed in particular region? Why not in all the places? Why don't you produce acids in your eyes? Yes.
Ma'am, is it because the protein, the histones get bound to a particular this only segment only to form the gene, the chromatins?
No. See,
Or
no,
it is
nothing to
because
do with the DNA actually here, because everywhere the same DNA is going to be present, isn't it? Right?
no, I meant like the particular sequence is only decoded.
But how does it know that?
Every segment of a DNA or gene might produce a particular protein which acts on the maybe like the protein responsible for acid production to the stomach and for tear production to the eyes.
Okay. But where is that particular protein produced from? See, all the information is there in your DNA, but where is that protein coming from?
The genes code for the protein formation, ma.
Yes, that's right, Amrita. So I asked you how only tears are produced in eyes and stomach is producing the acid. So Asmita said like some protein or something is going to be triggering only that particular, yeah.
Is it because of the tRNA?
tRNA, no.
Transfer.
Oh, no. Kind of like, see, only when transfer RNA comes and binds to the mRNA, you're going to be getting the protein sequence. That's right. But it's not at that level. You have to think it a little before itself. So we'll be start. Okay, I'll let you know the answer. Okay, we don't have a lot of suspense. Yes, somebody's.
Ma'am, one last case.
Yeah.
Is it because as we have enzyme on the substrate, maybe the protein binding to the enzyme, that particular place will have attached binding molecules.
On the DNA, you say, on the DNA region, you say?
No, ma'am. In that particular place where it has to reach.
Okay. These are all.
Ma'am, it has nothing to do with DNA or that OTR region.
It actually does in that region only. I'll tell you what. So it's called as gene expression. So there we'll be talking about gene expression in the later part of your chapter, but I just want you to be knowing it before. Why? Because that will make you more interesting to read this chapter further. So when we start with a healthy discussion of this chapter, it will be very interesting for us. So it's the reason why I'm putting that here. So it's because something called gene expression. A gene is going to be expressed when there are certain molecules in the environment. So for example, if there is a bacteria, okay, so bacteria is there, the bacteria is going to be surviving in, if it's surviving in that environment, it has to have the ability to produce certain kinds of enzymes. So it's because of the environment which triggers only that particular thing to happen. So what is in that environment is more important. As we were all very close to the answers, you, Asmita said it's like the something protein that's present in that particular region might, yes, it's called as a repressor protein. It's called as a, there are lots of things. Hi, Varuna. You're late to class by 10 minutes.
I thought we didn't have class today. It was actually for the next week.
Yeah, next week. Oh, you didn't want class today also?
Oh, ma'am, I actually thought we didn't have class today.
Okay, fine. So next week.
So can you do like a quick recap of what I missed?
Yeah, we are just starting. This is the first slide I'm doing actually. So we are doing about this chapter called genetics, and we are just discussing about the basis of it. Okay. So yes, we'll get back to the discussion again. We'll go back and I'll just revise it at the end of the chapter. Yes. So that's what it is. It's called as gene expression, children. So it's like in particular region, if tears are to be produced, you don't produce tears all the time, but the lacrimal glands do have these secretions. So it gets out of that particular site only when necessary to wash the cells or do something else. So it's like that. So things are not wasted in the body. See, we eat a lot of food, we need nucleotides, we need a lot of things. So many things are required to make small proteins in the body also. So that should not be wasted. So everything need not be produced in every sites of the body. But remember, mucus is produced in the nasal region, in every openings in the body. Nasal region, the throat, the esophageal region, the stomach region, on all linings of the body. So certain things are triggered or certain things are required everywhere in the body. So we'll be talking about these things in the molecular basis of inheritance. But in this chapter, we are going to be majorly focusing on how inheritance is happening from one generation to another generation. We are not going to touch into the details of DNA or RNA or something like that, but that's going to be the next chapter. In this chapter, we'll understand the basis. Okay, so so genetics is going to be dealing with the inheritance as well as variation of characters from parents to offspring. Now tell me, why is variation important? Why today Sneha is very quiet? By now she would have given me some answers. Yes, Sneha. Okay. Why are you very quiet today?
No, I'm listening. I'm understanding the concept.
Okay, right. Okay, I just want all of you to participate in the discussion because I would know how everyone like I will know you only when you ask me questions, right? Hari, this is how we usually deal with the classes. I would like you to ask me questions now and then also.
Yes, ma'am.
Yeah. Okay. So this is what we're going to be talking in this chapter, the basis of genetics actually, right? So what is variation? What is variation? I told you inheritance. What is variation? What is variation? Why is variation very important?
Ma'am, to keep life continuing, it is important.
Okay. Apart from that, first of all, what is variation?
Subsequently.
What is variation?
Ma'am, when there is slight modification in characteristic when inherited from the parent to the offspring.
Oh, only the so usually children think variation means the changes in the faces or something like that. We don't resemble. It's not just the resemblance, children. So there are lots. Yes.
DNA replication.
Exactly. On the DNA itself, there are going to be few changes happening. Why is the changes happening? Because of some environmental factors or because of some exposure to different things, or naturally there is some changes. So that change, we call it as mutation. So we'll be talking about this also in the later part of the chapter where we'll deal about evolution. So variation is the basis of evolution. So we will talk about something called as survival of the fittest. So that survival, see, for example, let us talk about a scenario where a father is being working for a long time in a cement industry. So the father would have been exposed to a lot of pollutants for a very long time. Then he has a boy child or a girl child. So those children naturally will have resistance to that kind of pollutants. So they will adapt to that place naturally. Okay, they need not have any particular adaptation or acclimatization for this. This is a random chance. I don't tell it will happen, but it can happen because there would be changes in the father's DNA due to environmental factors. Okay, so that would lead into the children developing the resistivity or resistance to that particular pollutant. So these are all something that we will be talking in this chapter. And that is why variation is important. Variation will make us survive in that environment. See, if we take our great grandparents or like five generations before us and make them to live in this particular environment, they will not be able to sustain. Why? We are living in a heavily polluted cities now. Am I right? 5 generations back, there was only agriculture, there was only bicycles, there were no pollutants, there were no industries, nothing was there. So those people's or those ancestral DNA, those 4 great-great-grandparents' DNA will not have the genes for surviving in this environment. So slowly we are able to adapt to this era because our parents, our great-grandparents, grandparents, or all of them were slightly started to expose to these environmental pollutions, and slowly we were also able to adapt. This is not a natural mechanism that we have already. So we are Homo sapiens sapiens. We are not the same Homo sapiens who were existing 1 million years, not 1 million, 10 million years ago or so. So we are Homo sapiens sapiens. We are subspecies of the human who existed 10,000 years ago or 50,000 years ago, because we are not the same. We have accumulated a lot of variation in our DNA, right? Okay. So this deals with the inheritance as well as the variation of characters from parents to offsprings. Yes. So this is really interesting. How does the elephant give birth only to an elephant? How does the monkey gives birth only to a monkey? Why the man only gives birth to a human? All these are because of, all this is because of what? The reserve. We have the DNA that is being intact. See, we have 23 pairs of chromosomes. That 23 pair of chromosomes, that's what meiosis happened. That's what we were discussing in the last chapter. When mitosis happened, the number of chromosomes would see what is happening. We have only 23 chromosomes in the gametes. So 23 plus 23 would give obviously 46, which is the human chromosomal numbers, right? It's a human chromosomal number. So if already it is 46 and this is 46, it would lead to a 92 chromosome containing human, which would be a different species, isn't it? If there is 46 set of chromosomes, then it is going to be a human. If not, it's going to be a superhuman or some alien or some other organism. It's not going to be a human. So this, what do we say? This number of chromosomes or the message is being preserved in the DNA. And that's what is happening during the sexual reproduction. The gametes are being formed. The gametes are only fused. The cells are never fused. You know, last week I just studied one, I just wanted to share with all of you. I actually have that. So they have artificially created an embryo without fertilization. I'll just... Okay, at the end of the session, I'll just share it with you. I wanted to tell every one of you. I asked you a question, if you all remember, why can't we fuse 2 female gametes to produce an offspring? I asked you a question and I told you the answer for that also. But surprisingly, I was just scrolling in the Science Daily website. I just saw a thing which was developed in the laboratory. So the human embryo has been developed artificially in the laboratory. So, but also it did not sustain after 3 months or so. It was not successful after 3 months because of certain reasons. Okay, I'll talk to you about that later. Right. So this is a rare photograph showing DNA relation. Person on the left is grand old father, and the right is grand old son, nearly more than 5 generations. Yeah. So see, this is what is the conditions that are being preserved in the DNA. So what is the DNA carrying? The DNA is carrying information for the eyes to be like this, nose to be like this. So all these materials is there in DNA. It is being preserved in the DNA, and the DNA is being transferring generation after generation. So what will happen? The great grandfather would have a set of chromosomes, which would have come to his son. So the same chromosome is there in the same genes would have been there in the son, then the grandson, then the great grandson. So generation after generation, the characters are being intact. They are being preserved in the yes, siblings are also sometimes closely resembling each other. So whenever you see two people, you can actually really point out this is brother and sister because of their similar resemblance, like their nose features, their eyes, and so on. So this is also a part of genetics where we'll be talking about things. So single gene trait in humans. Okay, so we are going to be looking into a lot of different things. So there are certain things that are being transferred from parents to offsprings. Only when the parent have that particular character, the children would have it. Usually this double chin. Here, when one of the either of the parents or the great grandparents would have had the double chin here, only then the child would have had it. Otherwise, it's not possible to run in that family. Either the father or the great grandfather or the people in the mother's family or somebody like that should have had that character. So it is a trait that is transferred from parents to offspring. This is another single gene trait in humans. Single gene trait means this is governed by only one gene in the human body. How many genes are there? So we have more than 22,000 genes. So human has approximately 22,000 genes. 22,000 genes that code for every function in our body. In all the 23 pairs of chromosomes.
Hitchhiker's song.
Yeah, I'll just show you the picture. We have the picture for everything. The clasping hands, the rolling tongue. So this is here attached. This is the hitchhiker's thumb. So few people can bend this here. I cannot do that. Maybe any one of you can. We usually have it straight. So some people can bend it back. That's not possible. So Neha is trying. So rolling tongue is really a great thing that people can do. All of you are trying, I guess. Okay. So the dimples are also.
Ma'am, what is clasping hands?
Clasping hands, yeah, I just want to say that. So usually see, when you clasp your hands, you can usually, you can put them together. A few people cannot just see the difference. I'll just tell you. So when they are putting the hands together, they cannot actually form what do I say? How do I say that? The position, see the position here on both these things, no? It's the superposition of each finger. Like when you have one finger on the other, we usually do this, but a few people see on this side, the one finger will be on the other side. We cannot do that. It will not be right for us. When you put this finger, when you put your left hand's finger outside, it will not be right, but for few people it will be. Someone else is trying. We usually cannot do that. We usually do like this, no? We clasp it. We put your hands together and clasp it like this. But this finger, so see here, no? Usually your right hand's finger will be on the left, but here, that will be the first finger here. But here, this will be the second finger here. Just try and see. You will not be, most of us will not be able to do that. It's like that for you, Neha?
Ma'am, yes, my hand is like the other one.
Okay, the opposite. Okay. So the left and right hand claspers. Actually, statistics says that 55% of the population are left hand claspers and the balance 45% are right hand claspers. So usually left hand, the dominant one will be left hand on this side, like this, what I do. But 45% of the population has it the other way. But I feel it is odd. I feel my hand is odd. But maybe for others, it's like, so that is clasping hand. Yeah, like 55%. So there is 1%, one or less than 1% of the population say they don't have any preference. Either way, I'm going to clasp 3 fingers. I don't have preference. But most of the people will be having a left hand claspers, and 44% of the other is right hand claspers. Right? Okay. So next is attached and free earlobes. So you know this earlobes, somebody's earlobes are attached. Most of us will be free. So a few would be attached. So is it attached, Neha, were you?
Yes, ma'am.
Oh, who's attached?
Mine.
Oh. Varuna, what about you? You're trying? Attached, huh? Free only. Okay. Okay. What about Sneha? Sneha is wearing mask, nothing I could see. Okay.
I'm free, but I'm not attached.
Sorry, Neha. Sneha?
Ma'am, I have free, but I'm not attached.
Okay. What about Hari and what about Sudarshan? How's your?
My also free, ma'am.
Okay, Sudarshan, yours? Sudarshan, can you hear me? He's again unmute. Asmita's is free, Amrita's is free. I can see it from here. Kiran Sadhana has covered her ears with her hair. I couldn't see anything. Right? Yeah. Okay. Right.
Only miss mine.
Okay. Okay. So head shakers, thumbs like this, rolling tongue, dimples. Okay. Father and his daughter are identical facial features. I actually thought this is fun. Then only I recognized this is not. I mean, daughter. Okay. So they have same facial features, etc. So when you come to these Northeast, no, a lot of people will have similar faces. We cannot identify them for some reason or other. They all have similar facial features. When you are very closely looking at them, only you will be able to really understand the differences they have. But usually, when you go to Northeast, like places like Assam and all these areas, Mizoram, and also you can see most of the people's features are going to be kind of similar. I feel that like they are not much mingled. So let's see, in India, when you take when you take people, a lot of people go here and there, they get mixed, the races mixed. There are lots of different people getting married, they get children and so on. So there are lots of different chances of getting different facial features, different variations more than similarities. But a few tribal population do not mingle with the other people. So their genes are kept intact. They are not mingling at all. So that is why a few in few areas you could see the similar facial features in them. Okay, these are also same photographs of okay, they look similar, kind of, not exactly, but yeah, we know about identical twins. So who's going to be telling me what are identical? Who are identical twins? How are they formed? We just saw in the last class. Except for the new person, anybody can tell. How are identical twins formed?
No, identical twins can be formed under 2 conditions. One is when the embryo is formed and later divided, and the other one is when rare cases where 2 sperms fertilize the egg simultaneously.
So that is monozygotic. So identical is monozygotic twins happen when a single egg is fertilized, then divides into 2, creating there is 2 different organisms. So you have to call it as monozygotic twins. I told you the term, I guess. Okay. So triplets are also possible when it divides into 3. There were some women who gave birth to 5 children. I remember recently quadruplets and finally, is there anything else?
Ma'am, what is the maximum?
I guess only.
Ma'am, because I read an article where they mentioned about 8 or 9.
8 or 9 is possible, but not identical ones.
Oh, okay.
Identical, I guess only 4 the max is possible. Because beyond 4, the division would be like, it would move to the next stage, the blastocyst stage or so, and it would just move to the next. I mean, not blastocyst stage. I don't think the space would be enough. We have word identical quadruplets only as of now. It's not possible, I guess. So what's this picture telling us? Oh, the similarities in all these trees. Okay, who remember what this tree is? Maybe, I guess only Neha was there in this class. Yes. Others should have not been. This was the 11th standards chapter. This is the tree in China called as the biloba tree. So it's a beautiful tree where when during the winter, it usually changes its color to yellow from green to shed, to get its own nitrogen supply from that of the leaves. So this is biloba. And why are there a lot of pictures? Okay. A beautiful picture of cabbage. If anybody wants to see how cabbage is grown, I guess they put that picture for this. Yes. So genetics not only deals with inheritance, but also deals with variation. So genetics deals with inheritance and variation. That's what we know till now. Yes. So what is variation? So we see non-identical twins. So what is they come from the same parent, they come from the same father and mother. So like siblings, they are non-identical twins are going to be siblings. So how do siblings come from the same father, same mother, but why are we different? Why are we different? That is what is variation. The degree by which the progeny differs from their parents as well as among themselves. So that's called as variation. What is inheritance? Inheritance is a process by which characters are passed from parents to progeny. So inheritance is a process where the characters are transferred. So how it is transferred? You all know about it. So by means of gametes. So we have already studied that. Now we are going to be learning the mechanism, how they knew this was only happening. How did we know that only gametes are transferred and not cells are transferred? How did we know that gametes are half of the, yes, Varna?
Identical twins, ma'am. Will they have the same type of fingerprints, ma'am?
Fingerprints would match, I guess, around 99%, but there'll be a very small change because of, I've read it, but they would not have the similar fingerprints. But when you see from a gross fingerprint would be same for both of them. But when you go into details, they'll have only 99.5% match and 0.5 would be different.
Yes, ma'am.
Yeah. They would have, okay, now the questions, will they have the same DNA? Yes or no? Will they have the same DNA? If yes, why yes? If no, why no? Kiran Sadhana has went to search the answer. Yeah, tell me. They would have it. Again, it's not going to be 100% same, but most of them will be same. Why? Because they share the genetic material from the same parents, same egg, same sperm, nothing, no change in it at all. So usually these twins would have most of the genes very similar to each other. That's why the facial features and everything are same. So mostly 100% the DNA would be same. If the DNA is same, obviously the fingerprint also would be matching. Yes. Why? Because even your fingerprints between fingers are different. So obviously they'll have a different fingerprint. Right? Okay, fine. Yes, coming back to this chapter, inheritance is a process by which character is passed from parents to progeny. Variation is that where natural, okay, so variation that were naturally present in wild population of the plants and elements to selective breed and choose the organism that possibly seek. Okay, so children, like very long ago, we did not have these things to eat. So rice was not cultivated, and all these things were not domesticated. So do you all know what is domestication? First of all, when did domestication start? Maybe possibly your sixth standards first chapter would have been like domestication of animals. We know domestic animals. Why did we not domesticate lion and tiger? Why did early man domesticated cows? Why did early man domesticate goats, chickens? Why did he not domesticate lion and tiger? Yes, so domestication means making them grow very close to your area where they will be very helpful for us. So variation that were naturally present in wild population of plants and animals to selectively breed and choose the organism that possibly possess desirable characters. So what happened naturally is still happening naturally in lots of places. So there are lots of variations. Where is that variation coming from? It's coming from the nature only. Obviously, variation is due to the environmental changes. For example, there is a cow in India. There is a cow in America. So what they did actually, what did they do? They actually bred both of them artificially so that in the Indian weather, these organisms are capable of producing a lot of milk, not a high quality. We are not worried about the quality of milk, but the quantity of milk was more, 400 liters, 300 liters. So that was the idea. So the idea was to selectively breed these organisms together for a betterment of the society or population or something like that. So through artificial, this had happened naturally in the wild. Till today in wild, few plants get a few new varieties of plants, new varieties of butterflies are all coming because of the selective breeding. So the organisms, so if you all remember, we studied this in our plant chapter, outbreeding devices. If you all remember, in the first chapter, after pollination, we studied outbreeding devices. So the plant itself doesn't want to be self-breeded because of the why plants do not require self-pollination. What happens if self-pollination is occurring generation after generation?
Ma'am, inbreeding depression is there.
Very good. So it causes inbreeding depression, which means the ability of these organisms to reproduce, it becomes lesser and the quality is declined. So what happens? Inbreeding depression creates. So to avoid it, outbreeding devices were naturally present in the plants. Similarly, this is happening in the natural environment to produce new varieties that can sustain in the environment. So this man has also adapted and started doing artificial selection. Through artificial selection and domestication from ancestral wild cows, we have well-known Indian breeds called the Sikwal cows in Punjab, so which is actually the huge milk producer. So this cow produces the maximum amount of milk, but the quality is high. I mean, the quantity is high. You cannot obviously know its quality. Maybe it doesn't meet the standard, but it is good. Okay, not bad. That's it. Okay, so this is what man has done through artificial selection and outbreeding. So this is not been a recent thing. We well know about this person called Grigor John Mendel, who was said to be the father of genetics, who has performed all these experiments way back in the 1880s. So what did he do? So Grigor John Mendel, 1822 to 1884, is really called the father of genetics. So actually genetics has different branches. This is called the classical genetics or Mendelian genetics, where we only talk about a lot of Mendel's discoveries and his findings and so on. That is called as classical or Mendelian genetics. Then we have modern genetics. Then we have molecular genetics. We have a lot of genetics. Today, genetics is all about genetic engineering and biotechnology, which is the basis of biotechnology, I would say. So we'll be talking about that in the later part of the chapter. But anyway, this person is the father of genetics, of classical genetics, I would say. So the period is very important. Please note it down, 1822 to 1884. He was the first to demonstrate the mechanism of transfer of characters from one generation to the next. So really think, no, today we know a lot of things. How did this man, how was he fascinated about how, oh wow, maybe he had a lot of time. He didn't have a lot of work to do, no homework, no schoolwork, maybe no home cooking or nothing like that. Maybe he sat, he was wondering how is this happening. So maybe that's the time that he had a golden idea of to work on all these things. He was the first person to demonstrate the mechanism of transmission of characters from one generation to the next generation. So we know this is happening, but how is it happening? The mechanism behind it was demonstrated by this person called John Mendel. What did he discover? He discovered the basic principles of heredity through experiments in a very simple plant called the pea plant. So really, this man is a genius, actually, I would say. Why? Because he has chosen a wise plant to, why did he choose those plants? We'll be talking about that in the next few slides. And that was his experiment. He has discovered a lot of important things that is today the basis of basic things behind a lot of experiments that are being carried out till today. Okay, he published the results of the studies under the title Experiments on Plant Hybrids. So today we commonly, lot of things, whatever we eat today, a lot of things are hybrid. Nothing is wild or purebred. Okay, so what is pure breed? What is hybrid? So have you all seen these watermelons that have a dark rind, smaller ones, dark rind, no seeds in it? So that is usually actually a hybrid. The natural ones are usually huger and they have lots of seeds in it. The seeds are said to be the babies of the plant. So that is where we get new varieties from. So if you are fertilizing without the seed, then it's going to be called as, what is that called? Fertilization.
Parthenocarpy.
Yes, very good, all of you. So I'm very happy all remember the other chapter. So the parthenocarpy is fertilization without seed production. If there is no baby formation, if there is no baby, then why do you need the upper part? That's not of use. So this is generally for, it's similar like the egg that we produce with the broiler eggs, which doesn't have any embryo in it, and just the new, what is that? The nutrient materials, like what's happening. Even I can relate like this. So no fertilization, but endometrium is there. Similar to that. So endometrium is going to be having all the nutrients in it for the baby to come and get into it, right? So you don't have the fertilization at all. You don't have the fertilization at all, but you have all the portion that's going to be producing or it's going to be useful for the seed to develop and so on. So that's called as parthenocarpy. So most of the fruits, vegetables or grains that we eat today are hybrids. Nothing is purebred actually. So this person was the first to start experiments on plant hybrid. So what did he do? For the first time, Mendel conducted hybridization experiments on garden pea plants for not 1 year, it's for 7 years. Okay, the period is again important. His period was between 1820, from his birth and death is 1822 to 1884. So his experiments were between 1856 to 1863, which was 7 years. Okay, he proposed the law of inheritance in living organisms. So he proposed the law of inheritance in living organisms. Sneha, are you supposed to go today also at 7:30? Are you, yes? Did you say anything? I couldn't hear you.
Yes, ma'am, at 7:30, ma'am.
Okay.
Have to go.
Okay. Maybe like I just told them, but I couldn't convey the message to Sajid sir. I will just tell him possibly this week. So next week we don't have classes, so that's fine. So the upcoming week we'll just make a different schedule if possible. So he proposed the law of inheritance in living organisms. So Mendel applied, yes. You know why this person was really successful in his experiments? Because he was the first person to apply mathematics to the experimental data, what he obtained. So when you read about this person's story, it's really interesting. We had, like when we were doing in colleges, we had the biography of Mendel so to read about what were the struggles he faced. People started laughing and he said, and they said mathematics is totally different and biology is totally different. You cannot combine both of them. That's not at all a possible way. People started laughing on his experiments and he was actually chucked out of the school and he wanted to work somewhere. He came back to monastery. Like a lot of things happened to him. And then only he was able to focus on so many things and do this. So Mendel applied statistical analysis and mathematical logics to problems in biology for the first time actually in the history of biological experiments. So that's why he was successful and that's why we still speak about him till today, right? So what is hybridization? I guess you all remember the artificial hybridization process that we studied in the other chapter, the bagging and emasculation. So that those were the two steps here also he followed. He did artificial hybridization. So what is hybridization? You are crossbreeding two different things. When we have to crossbreed two things, we have to keep two things in mind. We have to be very particular in choosing the variety of what we are going to crossbreed. So once you crossbred them, you have to be very careful. No cross contamination happens. So what was the first step? First step is emasculation. What is emasculation? If you all remember, what is emasculation?
Ma'am, removal of the stamens.
Yes. So is pea plant's flower bisexual or unisexual?
Bisexual.
Okay, right. It's a bisexual plant which has both male and female. So the stamens were removed, and what stamen? I mean, here you have the female part remaining in the plant, and the male part is being transferred to the female plant. Well, you can ask me, can't we do the other way? Why can't we do the other way? Why only the male gametes are transferred?
Because the female gametes cannot be removed. If it's removed,
Yes,
then that
right.
formation of pollen tube.
Right. Yes. I just want to know all of you are like awake or not. I may be asking these kind of stupid questions, but still, I'm just asking you to be in line with me. Right. So Mendel investigated several characters in the garden tree plant. When he was sitting idly, happily looking into the garden, he saw a lot of different things. He was saying, oh, why is the butterfly sitting only on this plant? What is the difference in this plant? What is the difference in another plant? He was wondering what were the differences. So he also noted many characters are manifested as two opposite traits. So trait and character are two different terms with same meaning. So please make sure if I say trait, if I say character, both mean one and same, right? So that's what, when he was happily sitting in the garden, he was looking into two different tree plants. One was short, one was tall, but both of them gave same fruits. One was having purple flower, one was having white flower. Then he was wondering how all these things are differing. So that was the basis of his intelligence. So the tall and dwarf plant, yellow and green seeds, etc. So what these were called as opposite traits. He noticed 7 different opposite traits in these plants. We will be talking about those 7 different traits, I guess. Yes. Okay. The tall and the dwarf plants, that is the tall and the short plant, yellow and green seeds, are few examples of this. The purple and white flower, axil and terminal flower, round and wrinkled seeds. Some seeds were kind of wrinkled with not perfect round shape, and others were like perfect round yellow and green. Then the what is the other? The part color was green and yellow. Yeah, so these were the 7 different characters that you observed in the plants. So when there was a cross between tall and dwarf plant, so when there was a cross between the tall and the dwarf plant, what had happened? This is not as small as this one that's mentioned in the picture. Please kindly ignore this picture. This is really wrong. This is not as small as what's shown in the picture. The dwarf is going to be in the ground level, whereas the tall plant is going to be really 2 feet tall or something like that. The cross between yellow and green seeds, what had happened, we'll be talking about it. So Mendel working with pea plant in his garden. This is a imaginary picture that has been drawn in so many books. Just give me 1 second. I'll show you that 1 second. Sorry, I'm so sorry that it was a small problem here. I'm very sorry too. Yeah. So so he was working with a garden tea plant, and yeah, we know about what he did. Just in a few minutes, I forgot what I was speaking. Okay. So these are the contrasting characters. Wait, let me finish up all the animations. Yes. So there were lots of contrasting characters. So for example, if you take two different pea plants, you can observe the stem height is either tall or dwarf. The color of the flower is violet or white. Position is axial or terminal. What is axial or terminal? The new flowers, if it's on the on the top, on the what's that, apex, it's called the axial. When the new flowers on the sides, it's called the terminal. Pod shape is inflated or constricted. Even now, if you get pea plants, you know, peas from the market, the full pods from the market, some of the pods may be flat, but still you can find the small seeds inside them. Some may be constricted, like constricted, inflated or constricted is, inflated is completely like this. Constricted is for every pod, there will be a constriction like this. This is constricted pod, right? So you can find both of them still now. The pod color is usually green. We don't see yellow. So when you see yellow, they say usually it is the ripened one or something. But there is a variety which is yellow also. So we could see the yellow in the dried form. We usually don't see yellow in the raw form. Actually, our parents don't put the yellow ones. They get only the green. So the seed shape is round or wrinkled. You can see the wrinkled. Even now, the seed color is yellow or green. So pod color, seed color, both are there. Okay. Yeah. So it doesn't always mean the yellow pod gives yellow fruit or yellow seed. That's always there. Okay, we'll talk about it. Oh, everything has a picture here. Okay, anyway. So see, this is the white and the violet flowers, position, axil, and terminal. This is what is the constricted pod. This is what is the inflated pod. Constricted is like this. Every seed has the constriction. You can see, no, here every seed is visible from outside. Pod color green and yellow. Seed shape is round and wrinkled. Seed color is yellow and green. That doesn't mean we have. Okay. So Mendel conducted artificial pollination, that is cross-pollination experiments using several true breeding lines. Okay. Now the question is, how did he know that was a true breeding line? What is true breeding line? I will tell you later. Just give me 2 minutes. I'll just tell you what it is. So he conducted artificial pollination or cross-pollination. Both are same. So what are the steps in cross-pollination? Emasculation and bagging using several true breeding lines. What is true breeding line? I'll just let you know. So true breeding means they have gametes of the same kind. They are not crossbred. They are true breeding lines. I'll just tell you. We'll be talking about a few a few terminologies here. I'll just let you know before we conclude this class. A true breeding line is one that has undergone continuous self-pollination. We say no, self-pollination leads to inbreeding, depression. So these kind of plants are ones which have undergone only self-pollination all of the time. Yes, homozygous. Yes, homozygous. Yes, shows that stable trait inheritance and expression for several generations. So generation after generation, they would have been producing same kind of flowers, same fruits, same taste, everything would be the same. So true breeding is one that has undergone continuous self-pollination. So what he would have done? He would have not so Mendel would have not trusted on the already available plant lines. He would have continuously allowed it to self-breed itself. And he would have selected those traits or those plants for cross-pollination. So Mendel selected 14 true breeding pea plant varieties as pairs, which were similar except for one contrasting character. Yes, now we are going to look into one monohybrid cross, children. This is called as monohybrid cross, where we studied already that there are 7 contrasting characters. So here Mendel has chosen 14 true breeding plants, 14 plants, which are having all the characters same except for one character. So except for one character, which is going to be different in both different plants. Okay. So we'll see what it is. This animation tells us. Oh, that's nothing to do with the topic here. I thought this animation is something related to peas with yellow and green. Okay. So advantages of selecting garden pea plant. Why did Mendel choose garden pea plant for his experiments? You would have done this in your 10th grade itself, but why did he choose this for? What are the advantages? So it's annual plant. What is annual plant? What do you mean by annual plant? There are three types of plant: annual, perennial, and biennial. What is annual? Biennials, perennials, and annuals. What is annual? Okay, what is biennial? What is perennial?
The duration, how long the leaves will be green, ma'am?
I'm sorry.
This period. Annual plants will have green leaves throughout the year or throughout their lifetime.
Okay.
Ma'am, is it based on the production of flowers and fruits?
Kind of that.
Flowering process happens once a year.
Yes, kind of that. So you have to tell it as a reproductive phase. So we know that the plants usually have the vegetative phase and the reproductive phase. So vegetative phase means it's growing all its other parts of the plant, and reproductive phase is when it is going to be flowering, followed by the reproduction and so on. So there are 3 kinds of plants where you could see annual plant. Annual plant will complete its life cycle in one, it will complete all the things, like from the plant development to the fruit production, everything in one season. So, and it will die after that. Okay, it will die after that. So that will take only one season. So for example, peas, like annual plant will complete its life cycle from germination to seed production within one growing season. There will be usually 2 growing seasons, 2 seasons. First 3 months would be vegetative growth, and the other 3 months would be reproductive growth and all these things. After that, so that will take 2 seconds. If it's 1 year, so usually biennials will be alive for two growing seasons. In one time, it will be growing completely. All the vegetative parts will grow, and in the next season only they'll reproduce and they will die. So similarly here also, in one growing season, from germination to reproduction, all these stuffs will happen in one cycle. Only once in lifetime, after they grow, they complete their life cycle, they'll die off. That is annual. Perennials are once every year they'll be producing flowers, like mangoes, like tall trees. No, they'll be producing flowers for every season. They'll be giving fruits every season, and they'll continue their vegetative growth. Reproduction will happen only once for them, but the vegetative growth will happen every year. I'm sorry, reproduction will happen every year, and the vegetative growth will be happening throughout the year in case of perennial plants. No, it doesn't reproduce throughout the year. Annual plants means they they will not they will be growing, reproducing, then they'll die. That's it. You'll not have the plant again, Neha. So, you know, onions are actually biennials. They'll grow for 2 seasons actually. So in the first season, all the vegetative growth will happen. In the second season, flowering followed by the other things will happen. So we usually don't keep the onions for a long time. We don't want the flower. So actually what's there in the bottom is not the seed, isn't it? Have you all seen onion seeds? Onion flowers, we know. Onion stems are used for what we call that. We use it usually spring onions. We say them as spring onions. No, that's the stem of the onion. All of you are blinking, I don't know. So actually onions are biennials. They grow for 2 seasons. First season, all the vegetative growth will occur. There only you will produce this onion. So what type of thing is onion? Is it a tuber, I mean. But why annual plants are chosen is like every growing season, new thing is going to be produced, no? Once they grow, they'll die, that's it. One minute. Maybe I'm wrong, but I'm for sure I know annuals are these things. Yeah, Sneha, please.
Okay, ma'am. Thank you, ma'am.
Yeah. I'm just sharing a new screen. Please let me. What, Amrita, today you're also very silent. Boring, ma'am, this chapter. Correct? So the difference between annuals, perennials, and biennials is plants come down how many years they live. Annuals live only 1 year. That's what I said. Biennials live for 2 years, perennials live for more than 2 years. So annuals, not years actually, generally the growing season we usually say. Like, yeah. So see here, annuals which complete their life cycle in 1 season. So usually peas are annuals. Biennials in 2 seasons. Like in 1 season, the vegetative growth will happen, and the other season, only the reproductive growth will happen. Yes, that's the definition, Neha. You're right. So it's a plant that completes its life cycle from germination to production of seeds within 1 growing season. Yes, you can tell it like that. What's the difference between perennial and biennial, annual? So perennial means every year. See, if pea plant has given its fruits, the plant will die. That's it. You cannot get anything new from the plant. No new flowers, no nothing will happen in that case. Okay. That's it. The plant's lifetime has got over. So if you take the seeds out of it, so like grains and all, no? How do we take all the grains? So grains and all, every year you have to sow new grains. So for example, rice, wheat and all, every year you dehusk it to get the grains out of it, right? Similarly, here also in case of pea plants, once you get the fruits out of it, it's not going to be flowering again. That's it. They'll have only 1 flowering season. After that, they may not be able to flower. That's because only they are after its complete vegetative growth, the reproductive growth of flowering will happen. That's it. Whereas in case of perennials, they can they can produce flowers every year at the same season. So some vegetative growth will occur every year by then. For example, mangoes are usually present during the summer. So we call the month of April, May as mango showers. They produce lots of flowers and the pollinators are active at that time. That's why we get a lot of fruits and so on. So, so you can see here perennials are native. Yes, they don't die, but merely restrict reproduction. Exactly. So perennial plants will not at all die. Whereas annuals would die. Once they have flowered for one time, they will die. That's it. So annuals, biennials, and perennials. Okay. So it is an annual plant that has well-defined characters. Can you see the slide back again? Okay, fine. So it can be grown and crossed easily. So if you have very small flowers, you know, it's very difficult for us to cross. Whereas these flowers are at least in the size that you can emasculate them, you can bag them, and you can cross them also. That is possible only in case of pea plants. That's why he chose pea plant for his study. It is a bisexual flower containing both male and female parts. So why did he choose a bisexual flower? Why not a unisexual flower, which will be more easier for him to get the desired characters? Why is it not? Why did not he choose a unisexual flower? Yes. Hari, any idea?
No, ma'am.
First class itself is boring, huh?
Not like that, ma'am.
Okay, fine. Yeah. So why not unisexual class? Why only bisexual class? What's the reason? He'll not get to true breeding line. Obviously, when the flowers are unisexual, they are going to be cross-fertilized. Obviously, they don't have self-pollination at all, right? Obviously, it's cross-pollination, right? When it's a unisexual flower, yes or no? You didn't all get my point, right? Why did he choose a bisexual flower? In case of bisexual flower, usually self-pollination is predominant, where the same kinds of genes are going to be present in the, like both paternal genes are going to be same, isn't it? They're going to be from the same plant. The same plant only has produced the male gamete, the female gamete. Obviously, the fruits are going to get the same characters. So whereas in cross-pollination, it's two different plants which produces the male and the female flowers. Obviously, it's a cross-pollination. And for sure, there would be variation in it, right? But he was interested to know why the self-breeding plant itself is showing cross-pollination. That was the reason why did he choose a bisexual flower containing both male and female plants. It can be self-fertilized conveniently. You can self-fertilize it also. Why is self-fertilization important? We are obviously talking about cross-pollination. Why is again self-fertilization important? To make sure there is a reserved seed set. We studied this in that pollination chapter, isn't it? Seed set, preserved, already reserved. The seed set should be there for sure. If it is self-pollinating or cross-pollinating, they want the baby to be formed, obviously. So for that reason, self-fertilization is a process by which seed sets will form for sure. It is a short life cycle and produces large number of offsprings. Every seed, every pea is capable of producing a new plant, isn't it? How many peas will be there in one pod? Approximately 5. Yes, kind of 5 to 6. So from one pod itself, you can usually normally produce 5 to 6 plants. Out of it, let's assume only 4 are generating, 2 are dying, or 3 is generating, 3 is dying. Still, if you sow more than 100 seeds, or for sure you would get around 80 to 85 plants, or 50 plants at least. So it produces large number of offsprings in one pollination or in one reproductive cycle. So these are the reasons why he chose pea plant for his study. First thing is, it's an annual plant with well-defined characters. What is well-defined characters? The contrasting characters that are seen. It can be grown and crossed easily. You can grow them and cross them easily. So it has a bisexual flower containing both male and female parts. It can be self-fertilized conveniently. It has a short life cycle and produces large number of offsprings. So these are the basic, or these are the advantages of choosing garden pea plant by Mendel for his hybridization experiments. Okay, so who is the father of genetics? It's Gregor John Mendel. What role does Carolus Linnaeus play? He's the father of what? First chapter. Yes, what is it? Nomenclature. Your mic is not working, Neha. Am I not hearing all of this? Kiran Sadhana, are you speaking something? Oh, I'm not able to hear you. Is my voice audible to all of you? Can you speak just? Somebody speak now. Oh, it's not unmuting, huh? Okay. Just try to unmute now.
Yes, ma'am.
Oh, why did it happen? Okay. So, okay, fine, Neha. Okay, fine. So it's nomenclature. Okay, Anton van Leeuwenhoek is the father of microscope. Okay, fine. Gregor John Mendel conducted experiments in brinjal plant. None of these plant, tomato plant, the first pea plant. Okay. On garden pea plants. What is the name? What is the biological name of pea plant? Anybody?
Pisum sativum.
Very good. Pisum sativum. We'll be using that term next from the next thing onwards. So who proposed the law of inheritance in living organism? Gregor John Mendel. Okay, fine. Contrasting character. Okay, all these questions are very basic. I know all of you will answer. Okay, so did you all receive the PowerPoints last week? Because the mail was not proper. I guess you would have not received it. Probably I've told them, probably you will receive it this time, I guess so. Right, okay. So anyway, just a second. We have just started this chapter. We have not done anything with the chapter. I know we'll be meeting the next week after that. So all of you make sure you're in line. So we'll be meeting probably 10 days after this. So we would be doing a lot of problems from next class onwards. So I just want all of you to have a paper and pen with you. Siddharth, I didn't see you taking notes today. So it's mandatory to take notes because we'll be mentioning key points in this and so on. So I would request you also to take notes, Siddharth. So probably you would have been given instructions from the institute, but anyway, please do take notes also. Right, okay. So question. I thought of starting with monohybrid cross, but anyway, I'll just give you a gist of what is monohybrid cross. We have just 5 minutes more, but anyway, so here we'll be talking about, as I said, Mendel chose different plants with only one contrasting character. Sorry, I'm just doing the same PowerPoint again. Oh, can you all see the screen? No, okay, it's not visible from me. Okay, fine. So I'll just give you the gist about inheritance of one gene, so which is called as the monohybrid cross here. So we'll be talking about a lot of terminologies in this chapter. So I'll be talking homozygous, heterozygous, dominant, recessive, first filial generation, and so on. So What are those terminologies? In the next class, the first slide would be the different terminologies, what we are going to be talking about. So if I say dominant, if I say recessive, so what is dominant? Dominant is nothing but the ability of a character to express itself. So there are two different characters where one is going to be expressing itself. It has come before and it's expressing itself. It's called as a dominant character. Why do we call the other one as recessive? Because this has dominated it. It is not able to express itself. It is there, but we don't see it. It is expressing, but the dominant one is masking its effect. See, for example, we have two characters, tall and dwarf. So if the dwarf is trying to express itself amidst the tall one, already the tall gene has been expressed. The plant is going to be taller. The person is obviously going to be tall. Even though if the dwarf is expressing it also, it is not going to be relevant. Already the person has become tall. Even you express, he cannot get down back, isn't it? He cannot become dwarf again. He cannot become short again. Similarly, in case of plants also, the expression for tallness is already expressed, or the tallness has been, it has become, the plant has become taller. Now it cannot shrink to become dwarf as the small gene, the dwarf gene is expressing. So even though it is expressed, the dominant gene is masking its effect. It's dominating more. That's why we call it as a dominant gene. So we'll be talking about dominant, recessive, then we'll be talking about homozygous and heterozygous. Apart from that, we'll be talking about the first filial generation. So I'll just explain what's homozygous and heterozygous. So homozygous means, so when, let's assume there is a character which is going to be the black hair or black eye. So with both the parents have, so let's assume both the parents have black eye. They have the genes for black eye. So they have given the same gene to the child. Okay, so the child has, so we, before all these things, I forgot to tell you about what is an allele. So do you know what is an allele? We have gene and we have allele. What is an allele? What is allele? So it is allele is said to be nothing but it is an alternate form of a gene. So for example, if a person has a black eye, if a person has a character for black eye, so all of you know you have a pair of chromosomes. You have a pair of chromosomes. Let's assume this is the first chromosome, the position, third position. It is coding for black eye. It's coding for black eye. Okay. Same place, there is another gene that in the same place you will have one more gene from the father or from mother. Let's assume this is from father and this is from mother. This is from father and this is from mother and father. Okay, so here you have genes for black eye. Here you have genes for brown eye. So this is called as a place. Oh, I'm not sharing any slide actually. I'm just sharing a whiteboard. Can you all see the whiteboard? Oh, can you all see the whiteboard? Varuna?
Yes, ma'am.
Yes, ma'am.
Okay. Oh, Neha, you can't see it? Oh, maybe. I don't know. Right. Okay, just no, I will just wind up the class in a few minutes. I'll be talking about the same thing again also, don't worry. So if the father has the gene for, you have chromosomes in that particular chromosome, you have gene for black eye, you have gene for brown eye. So here you have two different characters. So allele is an alternate form of a gene where brown is one character, black is another character. So these two are said to be contrasting in nature. So that is what we call as an allele. Allele means an alternate form of a gene. So we'll be discussing in detail about these in the upcoming classes, which is going to be the week after that, and we'll try to probably complete this chapter, at least monohybrid and dihybrid crosses in that class. Okay. How are big heart suiting man? So yes, oh, did you study any research? Like usually, earlier days, the grafting were done. So usually they do grafting of eyes, grafting of heart from other animals just by making it suitable. So we share a lot of things common. So children, the question was Neha asked me, how does the pig's heart suit us? How do they transfer? Like they usually graft different organisms heart to human. What's the function of a heart? It's just for circulation. So what should it do? It should pump it. It should have its own pace to do other things. So, the goat eyes were also earlier grafted. So what happens is how it's not suiting. We are making it suit to the environment. They usually what they do is they'll suppress the immune system for some time using drugs called as immunosuppressors. So what the immunosuppressor will do is it will not be rejecting the graft. So we'll be talking about grafting in immunology, I guess, in your, do you have this chapter called immunology? I don't think so. Maybe in 11th standard you would have had it. So no, okay. So human health and diseases probably is immunology.
We have immunology, ma'am.
That's immunology chapter?
Yes, ma, in human health we have immunology.
Okay, yeah. So we'll be studying about immune system. Immune system usually thinks when something comes from outside as foreign. But there are some drugs called as immunosuppressors, which would suppress your immune system, which would lessen the macrophages that are circulating in the bloodstream, and it will reduce the immunity. So what will happen? The graft that you have kept will slowly adapt by getting all the blood. So normal blood would flow in and out of the heart, and it will become one of the tissue among itself. If the tissue start binding to each other, the graft cannot be rejected. So the person would be kept in a very sterile room with those immune suppressor drugs, where he will not get any other infection for around a week or two. Probably 14 days they will keep him under immunosuppressed drugs and in a sterile room until the heart fixes to its body. So we need some muscle to function for pumping the blood through the system. That can be a heart's pig or an artificially created heart, pig's heart, an artificially created heart, or just a pacemaker also. So it depends on what kind of malfunction is there in the heart. So if only the pacemaker is not working, if the pace is set by the sinoatrial node, we know that. So SA node is not working, then they keep an artificial pacemaker. If the muscles are not contracting and relaxing, if the muscles stretching ability has been lost, then only the heart is being replaced. If there are lots of blocks, if the arterial, coronary artery is blocked, if there are lots of different problems in the heart, then they go for a heart transplantation. Anything else, children? Yes. Siddharth, how was the class? I just require some feedbacks from you.
Ma'am, today's class was interesting.
Okay.
That's all, ma'am.
Okay. Okay, Siddharth. Next class onwards, I'll be shooting a lot of questions. Be prepared for that. Okay. I want all of you to be answering. Okay. Asmita, you're there?
Yes, ma'am.
Okay, fine, okay, children. So we'll meet on probably, what would be the date after that? Second, 22nd, 21st, right? So we'll be meeting on 21st probably. So then we would do this monohybrid cross, and we'll complete monohybrid and dihybrid cross. So all be in line with us, and I'll just try to send all the PowerPoints by tonight. Okay. If not, I'll mention the the institute to send you all the PowerPoints. Okay. Thank you, children. Have a happy week ahead and happy holidays again. Bye.
Thank you, ma'am.
Happy Independence Day to all of you.
Wish you the same, ma'am.
Thank you. Bye.
Thank you, ma'am.
Bye.
Bye, ma'am.